Well, hello everybody, welcome back again. This week, or in this presentation, we are covering Chapter 7, which is kind of a companion chapter to the previous chapter, which was, as you'll recall, all about cell respiration. Here we're talking about photosynthesis, and these two processes are kind of companion processes because, as you've already discovered, the raw materials, the reactants for cell respiration, are the products of photosynthesis, and the reactants of photosynthesis are the products of cell respiration. So we have this chemical cycling between the two processes, which is something that we need to take advantage of, because it turns out that some of the chemistry that we talked about in the previous chapter, particularly the idea of reduction and oxidation, are just as relevant here in photosynthesis, because it turns out the photosynthesis is also a series of redox reactions, because it's all about moving electrons. Now, when we talked about moving electrons in cell respiration in the previous chapter, it's all about electrons going downhill and releasing energy that could then be directed to making ATP as the end product of cell respiration. Here, we're going in the other direction. So we're now going to move the electrons uphill. And of course, if you've ever gone uphill, you know that that's work, and that means we need to have some energy. And because the laws of thermodynamics that we've discussed in previous presentations make it really nonsensical to use our most common source of energy, ATP, to make ATP. We found that out in the last presentation, that really doesn't work. If we want to capture energy to make glucose, which is what we talk about as the end product of photosynthesis, but as you'll see, is probably not the best molecule to focus on as the direct product, but more of that as we move through this presentation. We need to have an external source of energy. Just as we saw, we needed an external source of energy to constantly top up our supply of ATP through cell respiration. So we need a constant external source of energy to make the organic molecules that are the end product of photosynthesis, to move the electrons uphill so that we can make organic molecules and store energy in the form of those electrons in the organic molecules. And so, as most people are aware, and hopefully that includes you, light is the energy source for doing this. And the question then becomes, if that is the case, what is the nature of light that allows it to be used as a source of energy for chemical reactions? And so we'll address that as part of this discussion. But we're first going to start with what may seem like a topic that is totally unrelated to anything that may have a connection to photosynthesis. And it's the fact that in many situations, we find that people are doing some strange things. And one of the strange things, and unfortunately, a lot of strange things seem to be associated with Florida. One of the strange things that was reported back in 2013 was that two men were stealing used cooking oil. And you have to ask yourself, what on earth does that have to do with photosynthesis? Well, remember that cooking oil these days is a plant-based fat. Oils in general are plant-based fats. And it turns out that with some careful manipulation, you can turn used cooking oil into diesel fuel that can be burnt by a diesel engine. And the notion of biodiesel as a source of energy for trucking has become quite widespread. And that means that used cooking oil has a lot of value these days. It's too expensive to buy cooking oil that hasn't been used, but once it's been used, it basically can't be used for anything else. And typically it would have been disposed of in some fashion. But now, because of the ability to chemically manipulate it a little bit and turn it into something that can be burnt in a diesel engine, it now has real value. By the way, just as an aside, Most people don't realize this, but when Rudolf Diesel, the man who invented the diesel engine, first was thinking about a fuel that could be used in a diesel engine, he was actually thinking of using peanut oil. That was going to be his original fuel. So it kind of makes sense that used cooking oil, once it's been cleaned up a little bit and some impurities have been removed, could actually be used as a source of diesel fuel. So much so that some people that you might know, or at least one person that you might know, has got into the business of biodiesel, and that's Willie Nelson. He has his own brand of biodiesel that is sold through trucking fuel stops. And I believe it's, at one point it was called Bio Willie. I'm not sure what they call it now, because it kind of has, yeah, we won't go there. But he is very big into manufacturing biodiesel and biofuels in general. And so what has this all got to do with photosynthesis? Well, remember, this is a plant oil. And plants make all of their own organic molecules by combining carbon dioxide and electrons from water together to carry out a reduction of that carbon dioxide to create organic molecules. I mean, that's the point of photosynthesis. And so we could take a larger view here and say that all of our so-called fossil fuels, whether it be oil-based from crude oil, or from natural gas, or from coal, or from peat, all of those are products of photosynthesis because they are the fossilized remains of plant material. And that is what gives rise to all of our fossil fuels. That's the reason why burning those is such an issue, because they represent one of the largest reserves of carbon on the planet. In fact, we say that the largest reserves of carbon on the planet are all the currently living life forms, which includes you, me, and everything else that's alive, and all the fossil remains in the form of the natural gas, the crude oil, the coal, the peat, and the other fossil fuels that are buried in the earth. Those are the largest reserves of carbon because we can trace all of those back to plants and photosynthesis. And so here's a thought to dwell on. Every one of the carbon atoms in your body, and there are literally trillions upon trillions upon trillions of carbon atoms in your body right now, and all the carbon atoms that have ever been in there, and all the carbon atoms that will ever be in there, have all got there as a result of photosynthesis, either by plants or by algae or by types of bacteria known as cyanobacteria. They've all entered into the living portion of the biosphere via photosynthesis. That's why plants are considered to be producers and why plants are at the base of every food chain and at the base of every ecosystem. Because it's the only way for carbon to enter into the biosphere. Well, maybe I should back up on that because as you'll see, there is an alternative minor way. And so maybe we should be a little bit more conditional here and say that the vast majority of carbon gets into the biosphere via photosynthesis. There is an alternative way. And in fact, it turns out to have a very important connection to photosynthesis because it's a process called chemosynthesis. And I'll talk a little bit more about that. The book really doesn't get into it, but I would like you to know a little bit about it because it helps you understand something that I'm going to show you about the equation of photosynthesis, because the equation that is commonly taught, including the one that's included here in the textbook, unfortunately, while technically correct, does not take into account what we actually know about photosynthesis, but more of that to come. So one of the reasons why we start thinking in terms of photosynthesis when we start thinking in terms of fossil fuels and biofuels is simply because fossil fuels and biofuels come from the same source. They're a product of photosynthesis. One way or another, they're all a product of photosynthesis. So when we start to think about the importance of photosynthesis, we need to immediately remember that every organic molecule that is naturally occurring that we are dependent upon, and again, all of our food, all of our fuels, because we use fossil fuels as our primary source of fuels, all of those are the product of photosynthesis. And life as we know it could not exist without photosynthesis. So it's clearly a very, very important biochemical pathway that we really need to pay attention to. And much of the time we just simply ignore the plants because they're there in the background. They're the green stuff in the background. We don't give them as much credit as we really ought to because, as I said, all life on our planet is totally dependent upon those, with a couple of minor exceptions, as you'll see. So when we start thinking in terms of photosynthesis, what we have to remember is that there are some basic ideas, and you can see them outlined here on this slide. Light is the energy source, and somehow we're going to turn that light energy, and whatever light energy is, and we'll need to talk about that in a little bit of the detail, into chemical energy. Chemical energy stored in the food molecules that are the end product of photosynthesis. The raw materials that we start with, as I mentioned earlier, are carbon dioxide and water, the product of cell respiration. And as byproducts, we have the organic molecules that are the starting point for making all sorts of organic molecules. So we need to talk about what that starting point is. And then, of course, oxygen. And we know that the vast majority of life, from what we talked about in the previous chapter in cell respiration, revolves around using oxygen as a reactant in cell respiration. It is a major reactant for cell respiration. And in the absence of oxygen, obviously, cell respiration is nowhere near as efficient or energy producing as it might be if there is no oxygen. Well, if there's oxygen present. If in the absence of oxygen, obviously, it's not terribly efficient. So again, photosynthesis is kind of important. And in fact, we could think of oxygen or the appearance of oxygen on our planet as a major changing point in the whole of life. We actually talk about in evolutionary science the concept of the oxygen revolution, because we know that the early planet did not have much oxygen, if any at all, in the form of oxygen gas. All the oxygen gas that we find in the atmosphere today is the product of bacterial photosynthesis, the cyanobacteria, or algae photosynthesis, or plant photosynthesis. And so, in fact, the origin of photosynthesis, or the rise of photosynthesis in terms of making oxygen, must have had a huge effect on the trajectory of life's evolution. Because up until that point, everything was anaerobic. But as oxygen starts to build up, and we think the significant level of oxygen was around 2 to 3%. Currently, our atmosphere is about 20% oxygen. So about a tenth of what it is right now, was the key turning point in the history of life. Because at that point, the level of oxygen now starts to become toxic to the anaerobic life forms. And they have a couple of options, some of them not good. One is to simply move to an area where there is no oxygen, and some of them clearly did that because they're still around today. Some of them obviously had the ability to adapt, and all of us who take advantage of oxygen through aerobic respiration are the inheritors of that. The third option was probably the one that was most commonly played out, and that was to go extinct because you simply couldn't survive in the presence of oxygen. So photosynthesis historically must have had a huge impact on the history of life too, because without photosynthesis as we know it today, there would be no cell respiration as we know it today, because there'd be no oxygen in the atmosphere. So there are a number of really important reasons for understanding more about photosynthesis and recognizing just how significant it truly is and has been over the history of life. So we need to recognize that all organisms can be split into basically two different types of organisms. Autotrophs, which are those that can make their own organic molecules, their own food molecules, and the term autotroph, as you can see it written here, means self-feeding. So this means self-feeding. The alternative is heterotroph. And the implication here is that you need to get carbon, and I'll put in parentheses here, organic molecules. From other organisms. And this is what we do. And this is basically what most life forms do, unless they're photosynthetic or, as I mentioned, this tiny little group that do chemosynthesis rather than photosynthesis. So those that rely on light energy, in other words, they do photosynthesis, are then referred to as photoautotrophs, meaning that they not only can feed themselves, but they get their energy from light. So when we talk about chemosynthesis, we actually refer to them as chemoautotrophs, meaning they're capable of feeding themselves, but they get their energy from chemicals. And it was an understanding of the chemoautotrophs back in the 1930s and a recognition of exactly what sort of chemistry they were performing as chemoautotrophs that led to the final understanding of exactly what photosynthesis was all about. Because the two processes share a lot in common. They're very much parallel. When we talk about the equation for photosynthesis, I'll show you just how much they have in common. So we are going to primarily focus clearly on the photoautotrophs in this chapter because they're the ones that do photosynthesis. So here are the organisms that you would recognize that carry out photosynthesis that we would correctly label as photoautotrophs. So you've got plants, and we often refer to these as terrestrial plants, but there are aquatic plants as well. So you've seen plants growing in lakes and rivers and streams and the like. Then we have algae, which are almost all aquatic. Some live in the sea, some live in fresh water in lakes and rivers and streams. And then if you had an opportunity to break out a microscope, you could also take a look at the photosynthetic bacteria known as cyanobacteria. And as we have seen before, there is this idea called the endosymbiotic theory of the origin of chloroplasts and mitochondria that says that chloroplasts started out as cyanobacteria that eventually were captured by an early eukaryotic cell that already had a mitochondrion, a captured aerobic bacterium, and that this eventually evolved into the chloroplast as we know it today. So in fact, there is a strong connection between the cyanobacteria and the algae because they use chloroplasts, and plants because they use chloroplasts as well. So not surprisingly, when we look at photosynthesis in cyanobacteria, we see a lot of chemical and genetic similarity between the way cyanobacteria do photosynthesis and algae and plants do photosynthesis. So this is, again, one of the supporting pieces of evidence for the endosymbiotic origin of chloroplasts. So we do have all of these organisms using essentially the same biochemical process. And we'll identify how that process works as we get into this chapter. So in terms of where photosynthesis occurs, let's go straight to the diagram. So if we take a look at a typical leaf, you notice that there are layers of cells in the leaf, and we refer to these cells as mesophyll cells. Again. I'm not happy with that. Let's erase that and see if we can do it a little bit neater. So in the plant world, and just so that you know this, I think I may have mentioned this before, but if I haven't, I'll mention it again now. I'm a plant scientist myself. I really enjoyed working with plants when I was actively involved in research, and I still have a strong interest in plant science research. Technically speaking, I identify as a plant physiologist, which means I try and understand the chemical and biochemical processes that go on inside a plant, and also how a plant develops, how it gets its structure, and all sorts of interesting things like that. I'm interested in what goes on inside plant cells. So in the case of photosynthesis, this is something that I have a great interest in. So this is part of the reason why I really enjoy teaching this chapter. It's also part of the reason why I'm going to give you a little bit extra information that the book doesn't provide, but which I wish it kind of did, because I think it makes photosynthesis a little bit more interesting, at least from my perspective anyway. So once we look through a typical leaf, or look inside a typical leaf, what we find are these multiple layers of cells that contain chloroplasts. So the term mesophyll simply means a cell that has chloroplasts and therefore does photosynthesis. And you can see that there are two basic types, or two basic layers of cells in the cross-section of the leaf. So we've got the cells here that look like they're standing on end. These are called palisade mesophyll because they look like an old log fort wall. So if you look at one of the old forts that were made out of logs, the logs were stuck in the ground, the top was sharpened to keep people from climbing over the top. That's called a palisade. So apparently when these were first identified, someone said, oh, that's what it looks like, so let's call these palisade mesophyll. Underneath, there are cells that are more loosely packed together. It's more disorganized. And this is called spongy mesophyll because there are a lot of air spaces between these cells. And those air spaces are there for a reason. It allows water to evaporate into the air and come out through these little openings called the stomata. And the stomata are really important because they allow not only the water out, but they allow oxygen out from the leaves because we don't want to build up of oxygen in there. We don't really get into the reason why in this class, but there are really good reasons why we don't like a lot of oxygen inside the leaf. It turns out to be an inhibitor of photosynthesis if it gets too high. And then it also allows carbon dioxide in. And so there's always going to be a trade-off between how much water can you afford to lose versus how much oxygen can you let out, how much carbon dioxide can you let in. And in more advanced biology classes, we actually get into a discussion of what effect that has on photosynthesis and how plants have altered the way they do photosynthesis in some cases in order to be able to get around some of the limitations that trade-off places on them, because it can become a really huge deal in terms of where plants can grow and where plants can't grow. But ultimately, as you look around, you realize, well, plants can grow pretty much everywhere. There's very few habitats that plants can't grow successfully in. So clearly they've managed to figure out ways to modify how they do things in order to be successful in growing in different habitats. And it comes down to that trade-off between how much water can you afford to lose, How much oxygen do you need to let out? How much carbon dioxide do you need to let in? But that's for a more advanced class, so we're not going to get into that. Yes, I know I'm teasing. So be it. So once we look inside the photosynthetic cells, the mesophyll cells, we see these little green discs. Each one of those green discs is a chloroplast. And so there's about, on average, 30 to 40 chloroplasts per mesophyll cell. Then once we look inside the chloroplast, we find that there are layers of membrane. So this is what it looks like in the electron microscope. So this is an electron microscope image. And so when you start looking at the membranes, what you notice is that some of them, the membranes consist of stacks of little discs. Each disc is called a thylakoid, and a stack of these thylakoids is called a granum. So you can see the names here. There's thylakoid and granum. The plural of granum is grana, because there are multiples of these stacks. Now, when you look at a thylakoid disc, and it's a little bit difficult to see here, but if you look in some places, you can see that it's very true that these little discs of membrane have a space inside them, which is labeled here the thylakoid space. We actually sometimes refer to it as the thylakoid lumen, but lumen is simply a fancy term for space surrounded by a membrane. So that's basically what we've got here. So each of these discs has a space, but what is important is that each of the discs is connected to each of the other discs in the stack. So this internal space of each disc is actually continuous with the space of all the other discs in the stack. But it goes even further than that, because it turns out, and you can see that here in the electron microscope, here's a stack of discs. And if you look carefully, you can see that there's a connecting membrane over to this stack of discs. And it turns out that each stack of discs, each granum, is connected to the adjacent grana by these connecting membranes that also have spaces in them. So what it really means is that all of the thylakoid discs, and more importantly, the spaces inside those thylakoid discs, represent a large single compartment inside the chloroplast. But then there's a second compartment, and that compartment is the material outside of the thylakoid membranes. In fact, it's the material that the thylakoid membranes are embedded in. If that sounds like a term that we've used before, embedding things in something, then that's probably a good thing, because the stroma, which makes up the second compartment, is kind of the chloroplast equivalent of the cytoplasm or the cytosol. And given what we know about the origin of chloroplast, probably was in the original cyanobacterium. And it's in the stroma that the second part of photosynthesis, the reduction of the carbon dioxide to sugar, what's known as the Calvin cycle, takes place. The capturing of light energy, well, that takes place in the thylakoid membranes. And there are several large complexes that are involved in capturing light energy that are embedded in the thylakoid membranes. Now, we also know that energy from light is turned into chemical energy in the form of ATP and high energy electrons that are stored temporarily in a molecule called NADPH. Now, of course, we know that ATP can be generated using a hydrogen ion gradient. We saw that in cell respiration, and that hydrogen ion gradient was generated by the electron transport chain. Turns out that here in the chloroplast, it's exactly the same thing. Remember, in a mitochondrion, we have two compartments, the matrix and the intermembrane space separated by the inner membrane. Here we've got two compartments, the thylakoid space and the stroma separated by the thylakoid membrane. We've got the same setup as we saw in the mitochondria, and there's an ATP synthase in the thylakoid membrane, and there's an electron transport chain. All the pieces are there to make ATP. So we're going to see that. And it's going to be captured light energy that's going to elevate the energy of electrons to be able to provide the source of energy to make ATP, but then ultimately those energetic electrons are going to be used to reduce NAD plus to NADPH so that we have a source of electrons for doing the reduction of the carbon dioxide. So all the pieces of the puzzle for how photosynthesis can occur in a big picture are right there in the structure of the chloroplast. And the stacking of the thylakoid membranes can be explained by the fact that that's where the light energy is captured. If you stack the membranes one on top of the other, you allow the chloroplast to capture as much energy as possible, because any that gets missed by one disc could be captured by the next disc or the next disc or the next disc in the stack. It just allows you to capture as much energy as possible so that you can drive photosynthesis at the highest rate possible. So again, we have this connection between structure and function, something that we've seen over and over again, and we continue to see every time we start looking at processes. So that's the connection between the structure of the chloroplast and photosynthesis. The next thing we need to do is take a look at the equation, because the equation tells us a lot about how photosynthesis takes place. So here's the equation as it is given in the textbook. And it is technically correct. And you may have very well learnt this equation for photosynthesis before if you've ever studied this, perhaps in a high school biology class. The problem is that it doesn't really tell us accurately what the chemistry is all about. So the first thing that we need to do that ties us to the idea that we saw in cell respiration of reduction and oxidation. Remember when we talked about reduction and oxidation in cell respiration, we were talking about moving hydrogens around. And that was the equivalent in chemical reactions, or at least in drawing chemical equations, to moving electrons. So we know we're trying to energize electrons. The question is, where do those electrons come from? Well, if we take a look at the moving hydrogens, we see that the hydrogens are coming from water and they're winding up in glucose. The glucose has the carbon in them, so those electrons or those hydrogens must be added to the carbon dioxide to give us glucose. So if it's all about electrons, we know that water is the source of the electrons, but we shouldn't really put the arrow like that because that gives us the wrong impression. It's really that the water being converted to oxygen as the hydrogens are taken away, or more precisely, the electrons are taken away. So when we talk about loss of electrons, remember oil rig or Leo grr. Leo the lion goes grr. This would be loss of electrons, and loss of electrons is oxidation. So water is oxidized to oxygen. That means that carbon dioxide must be being reduced to glucose. And that is true. But this equation is still not correct. And the reason why it's not correct can be seen by looking at what we know goes on in a group of bacteria called purple sulfur bacteria. And purple sulfur bacteria do chemosynthesis. And their equation looks like this. Now, this was discovered by an American biologist named Van Nile back in the 1930s. And Van Nile observed that not only was this a redox reaction, but something that was related to photosynthesis was going on that explained something about photosynthesis. So here's what Van Nile figured out about the equation that the purple sulfur bacteria are using. So we have carbon dioxide. They don't use water, they use something called H2S, hydrogen sulfide gas, or sometimes called rotten egg gas. It stinks. Sewer gas, swamp gas or other names for it too. It stinks really badly. And in high concentrations, it's lethal. If you can smell it, you're okay. It's when you stop smelling it that you get into trouble, because it deadens the sense of smell in your nose. And that's when it's getting to lethal concentrations. So hydrogen sulfide, you can see it's got hydrogens in it. So this is the equation that Van Nijl figured out for the purple sulfur bacteria. And you can see that it looks almost identical to photosynthesis, with the exception that H2S is performing the role that H2O would performs in water, in photosynthesis. But notice, whereas we have 6 waters in this equation for photosynthesis, we have 12 H2S. And that gives us 12 sulfurs. And we get water as a product of the equation. But what Van Nijl realized was that all the electrons for reducing the carbon dioxide came from H2S. And the sulfur was released as pure sulfur. And if you ever look at a picture of a purple sulfur bacterium, you'll see little yellow blobs, globules inside the cytoplasm of the cell. Those are excuse me, those are globules of pure sulfur as an end product to what's going on here. Now, Van Nijl looked at this and said, this is a redox reaction. I can tell that. Carbon dioxide is being reduced to glucose, and the source of the electrons is the H2S. And so H2S is being oxidized to sulfur, and we get some water as an end product as well. It looks a whole lot like photosynthesis. And Van Nijl suspected that the proper equation for photosynthesis ought to look like this. It should be 6 CO2 plus 12 H2O gives C6H12O6 plus 6O2 plus 6H2O. And the reason why he made this, or drew this conclusion, is that he realized that all of the sulfur from H2S gave rise to the pure sulfur. If, in fact, the equation for photosynthesis and the process of photosynthesis is exactly the same, just substituting H2O for H2S, the implication is that the oxygen gas must be coming exclusively from water. And even though carbon dioxide has oxygen in it, none of that oxygen must be turning up as oxygen gas. The oxygen gas must come strictly from water as a product of the oxidation of water to oxygen. And that's why Van Nijl suspected that this should be the proper equation for photosynthesis. He just couldn't prove it. But subsequently, in the 1960s and 1970s, it became possible to prove it, because it turns out that oxygen comes in 2 isotopes, oxygen 16, which is the normal one, and oxygen 18, a little bit heavier. And you can isolate both carbon dioxide and water that contains oxygen 18 instead of oxygen 16. If you feed plants with oxygen 18 containing carbon dioxide, just as Van Nijl predicted, none of the oxygen comes out as oxygen gas. If, however, you feed the plants oxygen 18 containing water, the oxygen gas contains the oxygen 18. That oxygen 18, by the way, does not turn up in the glucose. It only turns up in the glucose if the oxygen 18 is in carbon dioxide. If the oxygen 18 is in water, then the oxygen gas contains the oxygen 18. So it's very clear from that, from Van Nijl's prediction and the oxygen isotope study that in fact, water is the sole source of the oxygen for the oxygen gas, which means if you are producing 6O2 and the equation says that you are, you need 12 oxygen atoms, which means you can't have 6 water molecules, you've got to have 12 water molecules. But having done that and accounting for all the oxygen, you now have a problem. You have too many oxygens from carbon dioxide. You only need 6 for glucose. You've got 12 in the carbon dioxide. So you've got 6 extra oxygens. And you only need 12 hydrogens for glucose, but you've got 24 here, so you've got 12 hydrogens extra. And so you've got 2 hydrogens for each oxygen. That's enough to make 6 waters. And so that's where these 6 waters come from as a product of photosynthesis. It is now chemically consistent. All the atoms on each side are present on both sides in the same numbers. And it now accounts for the oxygen gas coming from water and only from water. So this is the equation, and maybe I should box this in black just to point it out. This is the equation that everyone now accepts as the proper equation for photosynthesis. It's consistent with what Van Nijl observed with the purple sulfur bacteria. It's consistent with what the isotope studies show. It tells us exactly chemically what is going on in cell respiration. It still does not negate glucose as the primary product of cell respiration, of photosynthesis. It still leaves water and carbon dioxide as the reactants. It leaves oxygen and glucose as products, but it adds water as a product as well. It just works. It just represents what we now know is going on in photosynthesis chemically. So this is the proper equation for photosynthesis as we know it today, as we understand it today. So I've already talked about this, the idea of the electrons going downhill in respiration and now going up from water to produce the sugar. We need energy to do that. So there are 2 steps that are involved, known as the light reactions and the Calvin cycle. So it's summarized in this diagram, which is a nice diagram as a starting point to understand the connections between everything. So here are the light reactions. This is the first step. This is where light is harvested. In the process, water is split. So you can see that water is split. Oxygen is released. The electrons then are used to reduce NADP to NADPH, and some of the energy of the electrons as they're moved uphill is used to drive the synthesis of ATP. The ATP and NADPH then serve as a source of chemical energy, the ATP, and a source of reducing power, the electrons in the NADPH, to drive the Calvin cycle. And in the Calvin cycle, we take carbon dioxide and we reduce it to sugars. We typically say glucose in the equation, but as you'll see, glucose is probably not the best choice. But the primary sugar molecule that is produced from the Calvin cycle can be used to make glucose and anything else that you want. The resulting NADP plus and ADP and phosphate are then recycled back to the light reactions to be reconstituted as NADPH and ATP. So in fact, the ATP, NADPH, and NAD plus and ADP and phosphate act as energy bridges between the light reactions and the Calvin cycle. So let me just put a couple of notes in here. Water is split. So that just gives us a couple more notes to let you know what's going on in here. So we're going to talk about each of these stages separately. So the last two parts of the presentation, because there are only 3 parts in this presentation. So the next part is going to focus on the light reactions. And then the third part, the last part, is going to focus on the Calvin cycle and how we do the reduction of carbon dioxide. The next part, talking about the light reactions, that's going to be the longest discussion because we need to talk about the nature of light. We need to talk about how light energy can be captured in the form of energetic electrons and how those energetic electrons can then be stored as chemical energy. So it's going to be a lot of discussion about the interaction of light with electrons and how that interaction can add energy to electrons. But before we do that, there is a BioFlix animation. And again, you can find the animation in the chapter resource page. And this is a really nice, about 3.5 minute animation. Most of the BioFlix animations are about 3.5 minutes long, approximately. It's a really nice 3.5 minute summary of the light reactions and the Calvin cycle and the role of ATP and NADPH as the energy bridge between the 2 parts of photosynthesis. Also tells you a little bit about the chloroplast, the grana, the photosystems that are involved in capturing light energy. Gives you a really nice overview of photosynthesis. So I'd strongly encourage you to take a look at this animation. And for those of you who like seeing things in motion as a way of learning, obviously the animation is a good way of seeing this as well. So, time for our first break. We've been going for a little while now. So as I did with the cell respiration presentation, I'm going to break this up into blocks separated by a concept check with an actual shutdown of the recording after reading out the concept check questions and then coming back and answering those questions at the beginning of the next recording. So here are the concept checks for this time break. What molecules are the inputs and outputs of photosynthesis? It should be fairly straightforward. Name the 2 stages of photosynthesis in the correct order. Kind of important to know which order they come in. What molecules serve as the energy bridges between the 2 stages? So whether you are going to take an actual break at this point or whether you're just going to click and move forwards with me to the next section, I'll see you on the other side of the break.